Hi everyone, and welcome to our video on our first class of macromolecules, the carbohydrates. Carbohydrate is a broad class of molecules that includes simple sugars that we refer to as monosaccharides, and the polymers that we can build from them, termed polysaccharides. The molecular formula of monosaccharides is typically a multiple of CH2O. So for every carbon, we have the same number of oxygens and twice as many hydrogens. The most common monosaccharide that you'll see in biology is glucose, C6H12O6. Before we move on to polysaccharides, I'm going to show you a couple of other monosaccharides, fructose and galactose. I'd like you to pause the video for a moment and see if you can figure out how these molecules relate to each other. Got it? These three are actually structural isomers. They all have the same formula, C6H12O6, but the arrangements of the bonds are different, resulting in molecules with different properties. They can also be combined differently to produce different polymers. Joining two monosaccharides together will produce a disaccharide. Prefix mono refers to one, so in this case one sugar, while the prefix di refers to two, so two sugars joined together. This is going to happen through a dehydration reaction. And if you haven't watched it yet, I recommend reviewing the videos on monomers and polymers before going too far into the different classes of macromolecules. The dehydration reaction here is going to happen between the hydroxyl groups indicated on the image, where one hydroxyl group will combine with the hydrogen from the other, yielding a molecule of water. The remaining oxygen of the second hydroxyl group will covalently bond to the carbon from the other molecule, forming the disaccharide, which in this case is maltose, because we've linked two glucose molecules together. If we had used different monosaccharide combinations, we'd have made different disaccharides. For example, glucose plus fructose will produce sucrose, the typical white sugar that you probably have a bag of in your pantry. Glucose plus galactose would produce lactose, the sugar commonly found in milk. Regardless of the type of monosaccharides involved, the covalent bond joining monosaccharides together is called a glycosidic bond or glycosidic linkage. You may sometimes see these labeled with numbers, like a 1,4 glycosidic bond, which we have here, and that tells us which carbons are involved in the bond. Typically, the carbons in a monosaccharide are numbered, with number 1 being closest to the carbonyl group. The prefix poly refers to many, so a polysaccharide is many sugars joined together. In biology, the most important ones are all polymers of glucose, but even these differ in their structures and properties. Starch, for example, is predominantly used by plants as a means to store energy. The most common form is amylose, which is what you're eating every time you have a plate of French fries or a bowl of pasta. The animal equivalent is glycogen, which is commonly found in liver and muscle cells, and serves as a store of glucose to meet energy requirements in animals. You'll see from the images here that glycogen is highly branched in comparison to amylose, and this serves an important purpose for animals. You're probably familiar with the idea of the fight or flight response, the body's way of responding to a perceived threat. If you hear a loud noise or see something scary, your body prepares itself very quickly to either face whatever the threat is or run away from it. Both of those responses require energy for your muscles, and that is provided by breaking down glycogen. The advantage of having all of these branches instead of a single end means that enzymes can go to work on multiple points simultaneously, releasing more glucose at once than if we could only break off one at a time. The final polysaccharide that we're going to look at is cellulose, and this is actually the most abundant organic compound on Earth. It's a major component of plant cell walls, and the reason that it's such a strong molecule for building comes down to a very small variation in the glucose molecules that it's made from, which allow it to form rope-like fibers made up of chains connected to each other by hydrogen bonds. So you might be wondering how polymers of the same monomer can be so different. Well, the structural and functional differences between amylose and cellulose can be traced back to isomerism in the glucose. Alpha glucose has hydroxyl groups on carbons 1 and 2, and when the molecule folds into a ring, both hydroxyls are on the same side of the ring. This allows it to form the 1,4-glycosidic bonds that we saw earlier in maltose. And adding more and more glucose in the same way will result in the formation of amylose, which, as we mentioned before, plants use as a way to store glucose for energy. Beta-glucose, on the other hand, also has hydroxyls on carbons 1 and 2, but they're on opposite sides of the ring, one above and one below. This means that beta-1,4-glycosidic bonds result in every other glucose molecule being effectively upside down relative to the one before it. And this is where the hydroxyl on carbon 2 comes into play. Because some of the glucoses are oriented the other way, there are hydroxyl groups facing each other when the chains are parallel. And these can form hydrogen bonds with each other, in the same way that we saw with water molecules. Collectively, these hydrogen bonds provide substantial tensile strength, and this allows plants to use cellulose to form rigid walls around their cells, protecting them from both their external environment and pressure from their internal environment, as we'll look at in later sections. Thanks for watching, and please make sure to check out the other videos on monomers and polymers and the other classes of macromolecules.